As a result of the Earth rotating around its axis, we and all other life forms on this planet experience the 24-hour day-night cycles, that is life on Earth. Around-the-clock circadian rhythms, from the Latin words circa and dian which mean about a day, are remarkably pervasive and guide nearly all of the biochemical and physiological processes in humans, including neural, endocrine and metabolic pathways. They also dictate our behaviors, such as sleeping and eating patterns. The constant 24-hour rhythmicity is maintained by our internal timing system, the circadian clock, which persists even in the absence of exogenous sensory signals, like light or food. This system has evolved to help humans to both anticipate and adapt to changes in the environment, by synchronizing the built-in clock with external time cues. The rhythms, however, can be disrupted, and dysregulation of the circadian clock can be significantly detrimental, contributing to a wide range of pathological conditions, including metabolic syndromes, neuropsychiatric disorders such as anxiety and depression, premature aging, and even cancer, to name a few. So examining the intricate relationship between circadian rhythms and their molecular mechanisms is essential to understanding the underlying physiology and pathology and diseases, and thereby directing therapeutic intervention. Researchers have been trying to decode the circadian rhythms for decades, and a number of important papers have contributed to our current conception of the molecular biology underlying clock function. As revealed by genetics and molecular studies done in Drosophila and mice, the time-dependent behavior exhibited by living organisms throughout the day is the manifestation of the endogenous cyclic gene expression patterns in the body. Present in virtually every cell of the organism is a core set of clock proteins that act on one another as activators or repressors in interlocking feedback loops, with a master clock located in the SCN of the hypothalamus, coordinating and interacting with all other peripheral clocks throughout the body. Importantly, the architecture of the core circadian clock is highly conserved across species and in particular, mammals. So studying basic animal models, like mice, provides important insight into the mechanistic basis of human circadian rhythms. The circadian clock impinges on multiple gene regulatory layers to ultimately ensure proper oscillation of thousands of clock-regulated genes in a tissue-specific manner. Interestingly, the circadian landscape in a cell can be drastically different, depending on genetic and epigenetic conditions. Recent developments in high-throughput imaging techniques and chromosome conformation capture 3C-based methods, applied in conjunction with next-generation sequencing, has provided remarkable insights into nuclear architecture, spatial chromosome folding, and epigenetic regulation. Chromatin possesses a great degree of regulatory information, and provides a platform for the integration and propagation of various signaling cues to influence gene expression. The architecture of chromatin in the nucleus is organized over multiple scales. Beyond histone and DNA modifications, regulatory elements such as enhancers and promoters can communicate with each other over large genomic distances to activate transcription through enhancer-promoter looping contacts, which are organized into topologically associated domains, TADs, within the nucleus. The study of the circadian transcriptional regulation, and in particular, Rev-erbα nuclear receptor, has been the focus of the research team at University of Pennsylvania led by Michael Lazar, for more than a decade. In this recently published paper, they investigated the mechanism by which the circadian clock is linked to chromosome dynamics, revealing the role Rev-erbα has to play. They used a chromosome conformation capture Hi-C technique to identify the interactions of adjacent DNA fragments, and determine how DNA looping that altered such interactions changed over daily cycles. They generated three-dimensional contact maps between distant regions for mice liver samples at 5 a.m. and 5 p.m. to determine genome-wide changes in organization. They identified extensive remodeling of EP interactions within hundreds of sub-TADs, smaller units within TADs with high interaction frequencies, containing circadian genes, with expression peaks at 5 a.m. or 5 p.m. For example, the circadian NPAS2 gene exhibited increased intra-TAD interactions at 5 a.m., including extensive looping between the transcriptional start site and 4 non-coding regions, E1 to E4. To identify the mechanism and relevance of circadian fluctuations in EP contacts, they focused on cryptochrome 1, Cry1, a major repressor in the core clockwork. Here, the study also revealed correspondence between rhythmic chromatin interactions and rhythmic transcription, suggesting that topological transitions characterize rhythmic control of clock-dependent gene outputs. Analysis of chromatin interactions, complemented with mapping of Rev-erbα binding, demonstrated that Cry1 EP contacts were circadian, in phase with Cry1 expression, and antiphase to Rev-erbα binding. The team showed the mechanism of Rev-erbα action to involve the recruitment of nuclear receptor co-repressor complex, with histone deacetylase activity, and subsequent eviction of the bromodomain containing protein 4, and its partner, mediator complex subunit 1, factors implicated in loop formation. In summary, chromatin conformation analysis now provide a new view of the core clock regulation, and highlight gaps in our understanding of how the circadian system prepares organisms for changes in the environment. This study has revealed a new principle of circadian gene repression by Rev-erbα, that involves the dynamic reorganization of genome folding at circadian loci, and the mechanisms by which Rev-erbα loosens the chromatin loops, leading to circadian repression of transcription, are likely to apply to other clock proteins, whose functions are currently not as well defined.